Welcome to this particular lecture on antibiotics by different natural products. In this particular lecture, we'll be discussing the antibiotics effect of different natural plant products that can be a suitable replacement of antibiotics against different pathogenic microbes because the antibiotic molecules are gradually losing the battle against the pathogenic microbes. It is because of the fact that antibiotics take long time to develop and at the same time huge money is involved. But the moment it is developed, this drug gradually they lose their efficacy in no time. Now this resistance towards antibiotic by bacteria is not a new story. It was explained by J. and E. Ledebug in their famous experiment of replica plating. In this particular experiment, what they saw was initially in this particular plate, large number of microbes were grown and then they were taken on a velvet sheet and then they were transferred to a petri plate containing antibiotic or streptomycin. But what they found was that normally they were not exposed to streptomycin earlier, but even then some of these microbes could develop in presence of streptomycin. So what does it indicate? It indicates that they have developed spontaneous resistance against streptomycin, and as a result of which streptomycin cannot kill this bacteria. As you know, offense is the best defense. So certainly there is a particular enzyme called beta-lactamase which destroys the beta-lactam ring of the common viable antibiotic penicillin, as a result of which penicillin gradually lost their efficacy, and presently you know penicillin is hardly effective against any human disease. The mechanism of antibiotic resistance may vary from one bacteria to other, and these mechanisms will be discussed by my colleague Sudeshna.
Thank you. The causes of bacterial resistance include the misuse and overuse of the drug. Number one. Number two, the changed environment may develop bacterial resistance as well. And horizontal gene transfer among the resistant bacteria may develop more antibiotic resistance. Not only that, the use of the transgenic, the host as well as vector of the resistant bacteria may help to develop the bacterial resistance against the antibiotic. And the resistance against one particular compound may give rise to the resistance against the similarly related to chemical compounds as well. Let's talk about the mechanism of the antibiotic resistance. There are four mechanisms. Number one, that is the destruction of the drug by the enzyme developed. In case of penicillin, with the help of beta-lactamase or penicillinase, it is destructed and the beta-lactam ring forms the penicilloic acid. Number two, as we know that in case of penicillin, PBP, penicillin binding protein is the target site. But in case of penicillin resistant bacteria and MRSA, we can find the resistance is getting developed by the alteration of penicillin binding protein.
What is this MRSA?
MRSA is methicillin resistance Staphylococcus aureus. Right. And you can follow, sir, how the development of the altered site against the penicillin in case of penicillin binding protein. That actually gives the resistance against penicillin. Third one, altered pathway. As we know that from para-aminobenzoic acid, folic acid and nucleic acids are produced. But if it is inhibited by sulfonamides, then no possibilities to produce the folic acid here. But we can find that in case of sulfonamide resistant organism, it is no more produced from PABA or para-aminobenzoic acid. Like mammalian cell, directly folic acid can be utilized. Next, another different strategy, very important strategy rather, that is reduction or decrease in drug accumulation. That may be done by decreased permeability of that particular drug, or maybe introduction of some ATP-driven efflux pump to get rid of the drug from the cell.
Actually, on the basis of resistance in a particular bacteria, it can be a natural resistance or it can be acquired resistance. Now, natural resistance is seen in case of, say, gram-negative bacteria, where there is a outer LPS layer, and this outer LPS layer is actually preventing the binding of the penicillin molecule with the specific penicillin binding protein. So naturally, the gram-negative bacteria are resistant against the penicillin group of antibiotics. At the same time, the bacteria structure may get modified with the exposure to a particular antibiotic, like it is seen in case of certain gram-negative bacteria, where the outer LPS layer will be modified in such a way it gets branched, and as a result of which the specific binding site of the antibiotic will not be there, and thereby the bacteria will become resistant. But we should also discuss the different side effects, because antibiotics have a lot of poisonous side effects, and these side effects can linger in human and other system, which is definitely preventing the long-term use of antibiotics. And these side effects include gastric ulcer. So there will be ulcerations develop in the stomach inner lining, or it can be developing different anaphylaxis or allergic reaction. And this allergic reaction can be from running nose to severe respiratory disorder, or sometimes it can be itching or flushing in the skin, or sometimes it can be GI tract hypersensitivity, diarrhea, vomiting. Large number of side effects are there which actually prevent the use of antibiotic in different people. And there at the same time, some of these very important antibiotics like cephalosporin, like clindamycin, like the penicillin and semisynthetic penicillin, like the fluoroquinolones, they all induce diarrhea. And this diarrhea is basically due to the loss of the friendly microbes present in the intestine. This is the human liver, and long-term use of antibiotics can actually induce severe liver damage in terms of cholestasis, in terms of mitochondrial dysfunction, that is energy generation, in terms of drug bioactivation, in terms of hepatocyte hypertrophy, in terms of hyperplasia, in terms of fibrosis. There may be different modification of the liver structure, and at the same time there will be modification in the innate or the adaptive immune system, and this will result in the long run severe damage like cirrhosis of liver. And as I was telling, that there are large number of friendly bacteria which is present in our gut, and these friendly bacteria can be helpful in different ways. That is, they may be competing with the poisonous or the pathogenic bacteria for nutrient. They may be involved in bioconversion of nutrient. Sometimes they may be producing some growth hormone, growth substances. Sometimes they can directly antagonize each other, that is the pathogenic organisms destroyed by the friendly ones. And sometimes they may have the competitive exclusion of the harmful bacteria. So in one way, the gut is nicely protected by these different probiotic bacteria. So when we are taking an antibiotic, these probiotics will also be destroyed, and we are vulnerable to the toxic side effects, which definitely include diarrhea. But this particular history and the development of the secondary metabolite is also equally important. I think Sudeshna should tell this particular aspect now.
Now let's start with the alternative strategies of all those antibiotics that are artificially created. Now the strategies may include fudge therapy, but have some toxic side effects as well. So we have to go for phytoremediation, the phytomedicines rather, phytotherapy, which has some sort of health benefits. Now in case of history, we can see like folklores in epics, you can find there are so many examples of the phytomedicines, so they can be used for that. Now in the modern trends, modern perspective, our India is biodiversified. We can have several ecotypes of different plants, so that can be collected, and by different companies from all the sources, the herbal drugs can be made. Now from this picture, you can observe how the alkaloids, isoprenoids, flavonoids, polyketides, they are all the secondary metabolites produced by the plant. They can be extracted, purified and commercialized for the herbal drugs as well. Now when we are talking about the benefits, it includes the therapeutic benefit as well as economic benefit. As we know that the higher plants are taken as the source for antimicrobial drugs, but here the foliose lichen can also be taken as the source for antimicrobial as well as antifungal drug. Next, when we are talking about the economic benefit, all those drugs can help to earn foreign monies as well as they can be used as cost-effective things for our people, our own people. So turmeric paste, sandalwood paste, they can be given over the skin. From this picture, it is followed that the comparison between inhibition zone of natural and artificial compound.
So it is really a cost-effective, healthy living.
Yes, sir.
Yeah.
Sir, can you please tell us about the structure of all those major components?
Actually, antimicrobial compounds, as you have said rightly, is coming from different sources. So when we try to classify them, it is really a hard job. So a part of it I will be taking up, and the rest I think you will share.
Yeah, of course.
So the first thing is they are the phenolics. The phenolics and the polyphenols like the gallic acid, the ellagic acid, or the galloyl beta D glucose, these compounds, they have a natural inhibitory action on bacterial development or on the development of bacterial cell wall. At the same time, there may be the quinones, which are the ubiquitous quinones, the anthraquinones, or the vitamins, the vitamin K, which is already acting as a anti-hemorrhagic factor. So by that way, it is controlling the hemorrhage and at the same time preventing spreading of infection. And this is a unique slide which shows that the flavonoid, which is acting on the cell membrane, will ultimately allow the WBC to come out and thereby will check the growth of bacteria in the intercellular space. So in one way we can say it is not directly harming the bacteria, but ultimately it is eliciting the defense reaction by which the bacterial growth can be prevented in human body. And there may be the tannins. Tannin, you know, is a very useful thing which is there in the tea leaves, and they are also used in printing ink, manufacturing. So these tannins are antifungal. And a very common experience we have when we wrap up our camera with newsprint which contains tannin, the fungal growth can be prevented. So tannins can be a very effective way of controlling the bacterial or fungal growth. And the rest, I think, Sudeshna will be taking up.
Interestingly, sir, another polyphenolic compound is coumarin, showing the antifungal as well as antimicrobial activity where the pyrrole rings are fused. That is actually effective against Candida species.
Right.
Next one. So the essential oil, the fragrance of the flower is coming from all those terpenoid group of compound, monoterpenoid, tri-diterpenoids, there are so many. In case of camphor, in case of farnesol, menthol, they are all antifungal as well as antimicrobial drugs. You can follow the production of terpenoids in plant system as well.
So it is there in plastid as well as cytosol.
Of course, sir.
So it is two compartment production of terpenoids.
Of course, sir, you are right. Next, we can find from a specific petalocymustol that has the activity to act not only against microbes, not only against bacteria, rather than it helps to reduce the candida size as well. And that is actually extracted from prairie clover. Not only all those compounds like polyphenols are related to polyphenols, because the isoprenoids developed in case of terpenoids, you can find the polypeptide, that is fabatin. In 1942, first the threonine 47 residues containing amino acid from barley and wheat which were extracted and has potentiality to act against bacteria and fungus. And fabatin is also the related compound as well. The most effective ones are the alkaloids, but they have some toxic effect, so that in very minute amount they are used, and all those alkaloids can have toxic effect as well, but very much effective. Not only that, there are so many other compounds like quinine and their derivatives like spermidine, polyamines. There are so many other compounds are there which can be extracted from the plant and can be used, like isothiocyanide, glucosides, etc. And you know, sir, sometimes in case of Brazil, the acetylins and flavonoids, they are used as antimicrobial, anti-malarial drug rather. Sir, can you tell us about the few points about the assay systems?
Yeah. Actually, assaying this natural product will require high efficiency system. One of this is microbroth technique, where we have a unique small titer plate. Well, what is the use of this titer plate? You can use multiple antibiotic or multiple antibiotic like compound. You can dilute them and you can actually find out the growth of the bacteria or turbidity in one shot. At the same time, there may be this unique rapid turbidometric detection, which is a VTEC unit, where you can actually quickly see the turbidometric assay of different bacteria. There may be a fluorimetric assay, where also by fluorimeter you will see the microbial growth, or you can use a very common PCR or fish-based technique. With this Aladdin unit, you can quickly measure that what is the growth of the bacteria in a particular suspension containing the plant extract. And I think it is now the time to discuss about the assay of these natural products and their extraction.
When we are talking about the antimicrobial assay, first it includes the principle. As we know that the higher plant are the source for the antimicrobial drugs, but in some plants they are so pronounced so that we sometimes need not to purify them at all. Next, let's talk about the requirements. We need tea leaves, we need the holoranin seeds, clove oil, vitamin C tablets.
Holoranin means kurchi
Yes,
seed.
sir. Yes, you're right, sir. We need sterile water, ethyl alcohol, homogenizer, we need centrifuge machine, incubator. We also need some other things like beef extract, peptone. We need the sterile hood or laminar airflow, cork borer, sterile petri dishes. We need the spirit lamp, and we need other solvent needed to extract them off. When we are talking about procedure, it first includes the extraction of the material from the plant source. Number 1, polyphenol extract from tea. 200 mg tea leaves are extracted with gallic acid. Next one, holorinus seeds. They are taken as the source for alkaloids. Usually, alkaloids are extracted in common sterile water, but to neutralize the solution, the caustic soda or sodium carbonate may be given, which can be distilled, steam distilled after that to get rid of the solvent. But here, in case of these holorinus seeds, only the neutral ethyl alcohol solution is sufficient for extraction. Next one, green tea leaves. It was done according to the Moriguchi et al. So what was done? 100 mg of the tea leaves are taken and extracted in dimethyl sulfoxide is to methanol solvent. 1 ml of that, 1:1 ratio. Then after extraction, they are centrifuged at 10,000 rpm for 10 minutes. After centrifugation, 0.5 ml of the supernatant was taken and diluted with sterile water up to 1 ml. For vitamin C, only the tablets are taken, powdered and dissolved in sterile water. Next one, in case of clove oil, as we know that serve as the source for essential oil. Many parts of the plant like stem, flower, they can be chosen for the source. But here we are taking the concentrated source that is commercially available. Then after extraction, that must be purified with rotary evaporator unit speed vac. So by which first the solvent containing active ingredient was evaporated, but next in second round bottom flask which is attached to a chiller or condenser, again we can gain all of those solvent containing active ingredient. Next one. So we have to prepare the inoculum. First, to prepare the inoculum, you have to make the nutrient broth. In 250 ml beaker or conical flask, add 0.35 gram of beef extract and 0.5 gram of peptone. Then add 50 ml of water only. Now it can be heated gently to mix them. We have to stir them off. Next, after doing so, you have to adjust the pH with 0.1 normal HCl or NaOH. After that, make up the volume with water up to 100 ml. Then nutrient broth will be autoclaved at 15 pound per square inch pressure at 15 minutes. Now it is prepared to inoculate the organism. The organism will be taken as gram-positive Bacillus subtilis or gram-negative E. coli bacteria. Then after inoculation, that will be kept overnight at 37 degree centigrade. Next one, we have to prepare the stab. The stab will be prepared like nutrient broth, but extra you have to add 2% agar. That is 2 gram agar in 100 ml of water. Now in 20 ml of sterile culture tube, we have to pour them off and make the stab. Again, you have to autoclave at 15 pound per square inch pressure for 15 minutes. Then after that, you have to pour the inoculum, you have to pour the liquid agar in the sterile petri dish, allow them to be solidified, and after solidification, you have to take 100 microliter of the inoculum, and with sterile spreader, you will spread the organism. Then after that, you have to prepare the agar wells. The agar wells will be prepared by cord borer by scooping off the solidified agar having 1.5 centimeter diameter. Now after preparation of the wells, extracts will be given. They will be in 20 microliter in amount. Now you have to keep in mind that in all aspects you have to add the control solvent, because solvent may have the toxic effect. So in case of vitamin C, only sterile water will serve as control. In case of clove oil, the neutral oil will serve as control, otherwise ethyl alcohol may serve as control. But in each case, sir, you have to perform the replicates for the statistical significance. Then all the petri dishes will be kept at incubator at 37 degrees centigrade, but in the upright condition. Next, we have to go for the observation after 24 hours by measurement of the zone of inhibition diameter, and we have to compare the test with the control. You can find here the study of solvent effect from this petri dish, which is serving here as control. Next, comparison between plant extract and control actually help to develop this particular table. So, name of the natural extract taken, volume or concentration of the active ingredient, if it is in the amount, so either may be microgram, if it is in the concentration, may be microgram per microliter or ml, then that will be correlated with the inhibition zone. Now as inference, it can be stated that lesser the zone of inhibition means the greater resistance. As precaution, we can say that every steps will be done under sterile condition. Always one control must be taken against solvent, and we have to avoid multi-drug resistant organism, because organism may be pathogenic, may not be, we should take care of, and all the plates will be in the upright condition, otherwise before diffusion they will fall out. Right. And we have to actually avoid the cross contamination, because we are extracting the plant extract, there may be all other components may present, so during extraction we have to avoid the cross contamination.
Should be sterile.
Yes,
the material should be sterile.
Of course, sir. And that can be crude extract or maybe most purified things, but we have to keep in mind that must be extracted from that particular plant part which gives the optimum yield of that particular substance.
So in this particular lecture, what we have discussed is, we have discussed the different demerits of different antibiotics. We have also discussed their toxic effect on human subject. And so we have discussed the importance of different natural plant product, how they can be extracted and how they can be bioassayed and how they can be used against the different pathogenic infection. Thank you.
Thank you.